for being here. Thanks to the speakers for their availability for this online workshop. It's called Open Cognitive Neuroscience, New Technologies, All Questions. It's organized by the Open Science Initiative of CIMEC, Think Open at CIMEC. Carlo Minucci, the director of CIMEC, and I, Vittorio Iacovella, we are part of the founders and steering committee of the Italian Reproducibility Network, a local Italian initiative to promote reproducibility in science. And this seminar is also, so the topics of this seminar are also related to what Italian Reproducibility Network is trying to promote. We started in a few minutes with a talk by Guiomar Niso and Rotem Botvinik-Netzer about open and reproducible neuroimaging from theory to reality. Quick housekeeping information: should you need a certificate of attendance, please write an email to thinkover.cimec@uniten.it. I'll write down this information in the chat as well when we are approaching the end of the workshop. For questions, we have one question and answer session at the end of each talk, and then a final open discussion after the two talks and the sessions. You can write down questions in the chat, or if you just write down question, we would call you on screen. We will check every time whether you are comfortable or not to appear on screen. So I think with that said, I can leave the floor to Guiomar and Rotem. Please, I stop sharing my screen and you can share yours.
Thanks so much, Vittorio. I'll share my screen. We're doing a joint talk, so we're gonna switch a few times between Guiomar and I. She will start, but let me share my screen first. I'm gonna let Guiomar take it from here.
Yeah, thank you. Can you hear me well?
Yeah. Yes.
Yeah. Okay. So thank you. It's my pleasure to be here today and together with my colleague Rotem at these Think Open Days. And thank you, Vittorio and team, for the invitation and organization of this event. In this talk, we will present our work on open and reproducible neuroimaging, going from study inception to publication and beyond. And this is one of our first times doing this joint talk, so please bear with us and for any inconvenience or any issue in the switching between us during the presentation. And also, I'm currently in a place of almost off the grid, so I apologize for any network connection or delays or breaking in the voice. Please let me know and I can try to stop, or Rotem can take it from there. Okay, so if you are attending this seminar, you will probably be familiar with open science practices and aware of its importance in science. However, as you may know, the adoption of open practices remains limited in researchers' day-to-day work. A recent study by Barrett and colleagues showed that 42% of researchers never pre-register a neuroimaging study, and 34% of them have never shared their raw neuroimaging data. So open science practices have many benefits for individual researchers and for scientific progress. They mitigate transparency and reproducibility issues. They increase exposure, reputations, chance of publication, citations, and many more things. So there are many new policies that are emerging by funding agencies to support also open science practice and tools. However, indeed, there's an abundance of tools, which is really promising, but also very challenging since the information is really scattered over many sources. And this can make it difficult even for experienced researchers to navigate this complex ecosystem of available tools and to make sustainable choices. That's why a group of over 20 researchers from different labs around the world actively developing and using open science tools in neuroimaging, join together, trying to provide an integrated overview based on our experience and on our expertise of these open resources available at each step of the research cycle. So we release a preprint at OSF that you can freely read, and if you are interested. And in this talk, we will try to summarize some of the main points. So in this work, we aim to provide, as we said, an integrated overview of community-developed resources that can support collaborative and open and reproducible neuroimaging throughout the entire research cycle and across different neuroimaging modalities, starting from study inception and planning, then data acquisition, research data management, data process and analysis, and finally dissemination, which comes back to generate new ideas and plan again a new study and start the cycle again.
Cool. So yeah, so as Guiomar said, we're gonna go through the research cycle stages, and I'll start with the first one, which is the inception and planning. And there are a lot of consideration there because it's really important to think about your study in advance and think how you're going to make it reproducible and transparent, because there are some things that are hard to do if you just think about them at the end. So it's really important to think about it when you start. So the first part is piloting, which is a very important part of research, but unfortunately, many times it's not done. But doing a pilot study is really beneficial because, it will increase the probability or the likelihood that your experiment will be successful, meaning that you can actually get the data and test the hypothesis that you wanted to test. So it's really important. It can be like with 1 or 2 participants, or it could be with a larger sample that you can use for power analysis and things like that. But importantly, what it helps you to do is to run all your experiment, make sure everything goes smoothly, the timing is as you expected, data are being saved as you need, and you can sync everything. You can sync the stimuli that you're showing the participant with the brain data, with any other data you're collecting, like physiological data or any other type of data. And make sure you're not, sometimes people just start collecting data, and then after 2 participants, they realize, wait, we didn't save some files, and we just need to throw out, for these participants. You really want to avoid this. So it ensures you get the data. It also helps you to get the data and then see if you can actually, run the analysis that you planned, if you get all the variables that you need, if, the assumptions, all, and everything around that. Sometimes piloting is not possible from different reasons. So some people use data that is already shared, like open data from other people using the same task, or you can also write some simulations to create data just to pilot that you can do the analysis. Of course, it doesn't help with making sure, the timing is right and participants understand the instructions and you save all the data, but it does help with making sure the analysis works. So this is important too. The second thing that is important is to document carefully everything. And a key document in this thing is the standard operating procedure, or SOP, which is a document that ideally just describes all your experiment, from how you recruit participants to what you tell them when they come and what you run when, how you connect the devices, how you set up things before, what the instructions you give participants, what you do at the end, how you save the data, just everything that the experimenter needs to know. And this is really helpful for documentation and for transparency later, but also for making sure that things work properly, and that if another person suddenly runs their experiment, or there are different experimenters, they are all doing the same thing, and you don't have like an experimenter effect or something like that. These things always develop during the project. You can never foresee everything before you start. For example, if a device fails or something in the connection doesn't work, you can add a picture of how it needs to be connected, or if participants do not understand instructions, you can change them and adapt them to make sure they do, etc. So it's really important to keep it alive and correct during the experiment, which can take some time. And ideally, someone can just take this document and replicate your experiment without any guidance from you. This is ideal. Obviously, it's sometimes hard, but the less they need your help, the more this document is well written. And also, ideally, this could be released with the data set or the publication or paper, because method sections have limited space, and we write as much as we can there, but sometimes it's not enough. And this document could really help someone understand what you did, or replicate it, reproduce it if they want to. There are examples of SOPs that you can use in other protocols in a few platforms, such as Protocol Exchange, Protocol IO, and Nature protocols. So I do encourage you to have these documents and share them with everyone as well. The next part that is important to do before you start the experiment is pre-registration. And pre-registration is the specification of a research design, hypotheses, predictions, and the analysis plan before you observe the outcomes of a study. Ideally, this is done before data collection, but it's also okay to do it sometimes after data collection, as long as you don't know the answer already, right? If you already know the outcome, there's no point in pre-registering because it's not pre anymore. And the idea here is to distinguish between two main types of research, exploratory research and confirmatory research. In exploratory research, we just explore the data, we torture it, we do a lot of analysis, just look for interesting things that can generate new hypotheses. While in confirmatory research, we already have an hypothesis before we look at the data, and we just use the data to test this hypothesis. And I want to make it as clear as I can. Both are needed, both are crucial for scientific progress, both are great. You need to do both, like we all need to do both. But it is important to distinguish between them, mainly because the interpretation you can make on each of them is different. So if data was used to generate the hypothesis, it cannot be used to test the same hypothesis. You need new data to test it, and it's important. And therefore, it's really important to distinguish between them. And the easiest way to do it is to do it before you have the data and look at it, because then you know that what you wrote in the pre-registration does not depend on the data because you didn't look at the data. So this is usually submitted to a public registry, and it has a timestamp. It's a frozen version, read-only, that you cannot change. Obviously, sometimes you do need to change something later, so you can post a new version of the preprint of the pre-registration and just explain what was changed and why, and people could see the first one and then the change, and that's all right, completely all right. But you cannot change the first one. This is the point, that it's there, and there's a proof of what you thought before you started, because we're human and we're very good at fooling ourselves. I study biases and inferences and human cognitive biases, and I know how easy it is to fool ourselves. So you want to just prevent yourself from doing that. There are three main platforms for pre-registration: OSF, the Open Science Framework, as predicted, and clinicaltrials.gov. There are a few others, smaller ones. You can use them too, but these are the ones that are most commonly used. I think the OSF is probably the most commonly used. It's the biggest one. You can also share data and code and other documents there. And there are many different types of pre-registration you can do there. There are many templates for, for neuroimaging, for example. There are some templates, and there are some simpler ones and some more elaborated ones. So you can really choose what works for you. Maybe if you're just starting, you want to start with a simpler one, for example, and not, dive into the hardest one. And so this is one great platform to use. As predicted is a much simpler one. It's shorter, much more straightforward about the details you need to give. So this is also a very good option. And clinical trials is, as you probably understand, for clinical trials, although the National Institute of Health in the US, the NIH, has broadened its definition of what a clinical trial is. But if you are doing a clinical trial, you want to pre-register it there because you need, in many journals, you need it for publication. So they won't publish a clinical trial if it wasn't pre-registered before. This is more strict. They don't have a lot of templates, a lot of flexibility. They know exactly what they want, and they want to make sure that you write everything. So this is one option to use as well. And there are many benefits to pre-registration. As I said before, it improves the transparency. It makes sure it's clear what you thought before you saw the data and what after, what your hypotheses were, the predictions. By doing this, it prevents most of the questionable research practices like p-hacking or erking. So just to make sure everyone understands that p-hacking is when you try a bunch of different analyses and selectively report only the one that worked or yielded a significant result. And erking is hypothesizing after results are known, which means you change your hypothesis after you already know the result of the hypothesis, which is kind of problematic. So pre-register, because you say what your hypothesis is and what you're going to do with your analysis beforehand, you're not likely to p-hack or do erking. Although, you can, but it will be transparent that you did, which is okay, because you can do exploratory analysis afterwards, as long as you say that this is exploratory analysis. And like I said before, this is great and needed in science. It also improves planning and helps spot mistakes before you start to collect data, because to do a pre-registration, you usually need to write something with all co-authors and make sure everyone are on board with that. So it makes it easier to see if there are any misunderstandings within the team. And also, if you plan your analysis, you can see, oh, wait, I didn't collect data about this variable, or maybe it's better to do this. And this is a great thing that you can do. And there are also some recognition that you can get these days for pre-registration, and many journals give you open science badges, and one of them is pre-registration. There are some prizes and some other things that you can do. So it also, like, if you're looking for personal benefits, maybe you can get them too. Taking pre-registration one step forward, registered reports is when you kind of do peer review to your pre-registration. So what you do in registered reports, first of all, there are hundreds of journals now that are offering this type of publication, and there are a lot of registered reports that have been published and are, with really high quality and are really changing the literature. So what you do here, you have your idea and you design your study, but before you go collect the data, you're writing your introduction and your methods. You write exactly what you plan to do and what your hypothesis is and what you think you're going to find. And then you submit it to the journal. It goes for peer review, which is about, how good is your methodological plan? How interesting is your hypothesis? Does this, methods actually test this hypothesis, and things about that? It can be improved, of course. This is a really great time to improve your experiment before you do it. And it can go for revisions. And if it is accepted, it's called an in-principle acceptance, which means that the journal says, as long as you go and now do what what we agreed on, and do your analysis, collect your data, do the analysis, and report the results, and come back to us, irrespective of whether the results are significant or not. This is going to be published in the journal, which is great because this is the best of science. Because if your hypothesis is interesting and you test it correctly, then it should be interesting anyway. Never mind if it's significant or not. But in other formats today, it's much harder to publish an insignificant result, a non-result. So this helps to improve science and make sure we're doing it better. And so then you collect your data, you do the analysis, and you write a paper as you would if it was a regular paper, and you come back to peer review stage 2. And then what they test there is not if you're, you have like surprising and cool results, but rather if you did what you said you're going to do, and if your conclusions are legit based on your results. And then if it is being published in this, accepted for publication in this journal. So this is really great. I encourage you all to try that. Another part of the planning and preparation is the ethics and sharing plan. So like I said at the beginning, there are some things that if you want to think about them and plan at the beginning, you will not be able to do at the end. So for example, if at the end you would like to share your data, you need to make sure that your participants agreed and consented to sharing their data before. And if you didn't do it, then it's too late at the end of the study. So you need to consider, so every country has different regulation. Europe has stricter ones these days. So you need to make sure you understand this and consider all these regulations and make sure that your informed consent, if you plan to share your data, then it indicates exactly what is going to be shared, where it's going to be shared, who will have access, and the participant consent to it. Okay. Usually what people do is to say they're going to share the anonymized data or de-identified data. You need to make sure it's clear, if they can later change your mind and tell you not to share it, can you take it back, things like that. And to help you, there are really, really great templates and a lot of information in the Open Brain Consent project, which has different templates in different languages. I don't know if they have Italian, they probably do, but you should look. It could be really, really helpful. And they also consider the GDPR rules, so the European ones. So this is a really, really great source to look at in advance and make sure your consent forms are exactly how you want them to be. And the last thing I'm going to cover here in the planning is quality monitoring, so quality checks. This is not exactly before, this is during, but you need to plan it in advance. And because, collect data and unexpected events always happen. Something gets wrong, there's any problem. And the earliest you're going to detect it, the more likely you are to be able to fix it easily, and not just use a lot of data or something like that. So it's really important to put in place quality control checkpoints in advance. For example, to look for artifacts in the brain, to make sure participants are not moving too much. Maybe you need to change something in the test or in the scanner to make sure they move less, or tell them to move less. Make sure they understand the instructions. Just make sure that everything is working as you planned. And this could be done manually or with the software. There are some softwares to do it. Here there are some examples for MRI specifically. The most often used one is probably the MRI QC, which gives you a lot of different measures on the quality control of your images. There's also visual accuracy and quality. And you can do it also by hand if manually if you don't have too much data or you have time, although it's sometimes harder. And there's active line of work to try to do it more automated. I mean, not just give you measures, but also give you some rules or guidelines on who which participant to exclude based on what. But we're not there yet. So currently you can just get a lot of data and see your who is an outlier in some aspects, or you can compare to other studies and things like that. Another very important thing is positive control analysis, which could be very, very beneficial to prevent unintended p-hacking, I would call it, because sometimes we do some kind of analysis and we see that something is not exactly right or assumptions don't hold, and we need to change, and then we're trying different things, and then we are actually p-hacking even if we didn't mean to. And we're human, so even if we really try not to, we're more likely to like the results if it's what we expected, or test it less if we get a significant one. So you should try to avoid it. And the positive control could really help optimize the analysis without seeing the results. So you can, for example, if you have a button presses, or if you have visual stimuli, you can make sure that you get visual activations or motor activations and try to optimize them before you look at your actual results of interest. So this could be really beneficial. And in some fields, again, like in pain study, there are some signatures that should be there for higher pain, and then you can make sure they are there and the signal is good, and then go to your outgoing filters. So this is one thing that can be very beneficial. And I'll go to the next stage, which is the data acquisition. This part is more technical, so I'll do it quite quickly, and then we'll go to the next part. So first of all, the brain data. One issue about open science is that most brain data acquisition devices or systems are vendored. So there's a company that sells it, and it's closed, and it's not open source. You can't see how it works, and it's different between different companies, or even different, for example, title scanners within the same company. So it makes a lot of difference because it could really get you some spurious non-biological differences across different data sets, and are not meaningful, but you can't really control. It's sometimes hard to control for that. But there are a bunch of people that are trying to actively work to develop tools to mitigate this challenge, and they develop open tools for that. So one thing the MRI community is doing is trying to find consensus protocols, like just a protocol sequence that you can use across scanners, but it's sometimes hard because the parameters are not exactly the same, and also you don't always know what's going on behind the scenes. And when you get the raw, what we call raw data in MRI, for example, the DICOM files, they're not really raw. Like a lot of things happen before you got this data. So sometimes we do consensus protocols. For example, in the Human Connectome project, they had a consensus protocol that worked across many different scanners of Siemens specifically. But there are some other acquisition sequences that are vendor neutral, not just for Siemens, but for all of them. That you can see, for example, PulseSec and PyPulseSec are softwares that can help you build such sequences. There are also some work on reconstruction frameworks that could be vendor neutral. And there are some tools that are being developed for quantitative MRI that could help you with this too. But for now, I mean, we can use all of these tools. And also it would really help to report in details your acquisition parameters and exactly what you did, so that people could maybe apply and understand what happened in other scanners if they want to implement it. So the reporting is very key here. And from the behavioral side, there are a lot of open source software actually to use, so this is really cool. And you can use PsychoPy in Python, or PsyCtoolbox in MATLAB, or other modality specific type like ERP Corp if you're doing ERP EEG. There are some like ReproStim for videos, and Pavlovia for some which is a repository and a platform you can launch some tests that are already pre-implemented by other people. So this is really helpful, and it's really beneficial to publish your to share your code along with your publication, so people could use it and do the exact same task if they want to replicate your study or do something else with it. And again, if you can't share it, or even if you can, try to describe it as much as you can, including the versions you use and software you use and everything, the computation environment. And I will let Guiomar continue.
Okay, so now we will talk about research data management in this section and the open resources that are available to deal with it. What's research data management? It refers to how data is organized, maintained, annotated, tracked, and stored throughout the research project. And in this part, we have divided into 3 sections. the data organization and standards one should follow to be more efficient, and then how you can add metadata and annotations, and finally how you can track your changes. So first, for the data organization and standards, there's a problem, and is that data is organized differently between institutions, and even within a lab. Different people organize the data differently in their own way. So this creates a lot of misunderstandings and waste of resources, and even poor reproducibility of resources. So having a standard to store, organize, and share neuroimaging data can facilitate that you can easily share between colleagues within your group, or between institutions, or openly, publicly in repositories. So it's really useful to have a standardized way of organizing data. So BITS stands for Brain Imaging Data Structure, and it's based on very simple file formats and folder structures. For example, here, if you see, you have your subject folder, sub1, then you will have your anatomy folder. Rotem, maybe if you can press. Yeah, sorry, it didn't show. And press a few times again, okay. Then you will have your data inside your anatomy folder, and your metadata together with your data. So this metadata gathers all the information related to your data. And for example, for MRI, it could have like a modality. You will state MRI, your magnetic field strength, your imaging frequency, your manufacturer, and all these details that have a specific field names that you have to follow, and that are documented in BITS specifications related to every modality. So this is very useful because these metadata files that are always together with the data are readable by humans and machines. So this can facilitate querying, curating, and all the documenting of your raw data. Also, there are multiple tools to facilitate using BITS and to incorporate BITS into current workflows of researchers. For example, BITS converters for the different modalities like MRI, MEG, EEG. This helps you to convert your raw data into BITS formatted BITS organized data. And this is sometimes a more difficult step, well, not difficult, but time-consuming step, but having these converters facilitates a lot the task. Also, as Rotem was saying, if you plan in advance that you want to share your data, then you start organizing it according to BITS, so then it's super easy. Also, we can provide you with a BITS validator, which allows you to ensure your data follows BITS specification. And if you have any error or any file that's missing, or any field that is not correct, then this will show you an error or a warning, and you can easily fix it. Also, we have some general tools like PyBits and BitsMatlabs that facilitates interaction with the data for querying, data plumbing, and manipulation. And finally, we have BITS apps, which are analysis pipelines, content arrays. So it's easy, like in Docker or Singularity, to reproduce your analysis. So because these apps always expect the data input in BITS form, organized according to BITS, then it's very easy to run and reproduce and to apply the same apps to different data. So this helps a lot in the analysis. Currently, BITS has many extensions for different neuroimaging modalities like MRI, which was the first that was created at Ross Poldrat's lab at Stanford back in 2015 and published in 2016, and led by Chris Gorgolewski. And then we joined with the MEG extension, and soon many other extensions started to also be merged, like EEG, intracranial EEG, genetics, recently PET and microscopy. And there are many more extensions under active development. So it's really encouraging if you want to also join this initiative, to go to the website and collaborate. They are very open and inclusive community, and you will feel very welcome. And maybe you can extend to the interest you have. So now we go to metadata and data annotation. So it's sometimes difficult to annotate your data because everyone uses their own words, and sometimes everyone follows different criteria. So without a standard, it can be a bit cumbersome and error prone. So that's the idea behind the hierarchical event descriptors, HIDs, which is just a structured vocabulary for annotating data, which is human and machine readable. This is integrated with bits, so and it can be also extended to any other research supply schema. So this is very convenient. For example, here we will, if we wanted to annotate this image, which is a face image, we can say, okay, this is a sensory event. All these things in the box are a structured vocabulary available at the HID. So it will be a sensory event. It's a visual presentation with an image, face, hair, and then the image has this name, and the condition variable is famous face. So you can describe many aspects of the image following the schema, and this is the short version. But there are tools that can extend this into and span the full path, allowing hierarchical relationships. So, sorry. So, for example, for the sensory event, you will say it's an event and then sensory event. So this long form allows you to have some relation between the categories and to establish more, to allow more analysis and metadata curation and querying and research. Then we have the neuroimaging data model, NIDM, which is a huge project and extends the W3 probe standard for neuroimaging, using provenance to link components of the different research stages of process of research. So through provenance, you can link each component. As you may see, they go through very basic level to a very more like a tangible level, from semantic web technologies to vocabulary, and then starting from raw data, how you, once you analyze it, you track your provenance, and then you have your pipelines, and then you have your results. And inside the NIDM terms, in the NIDM project, there are many other projects. And here we would like to highlight the NIDM terms, which collects community developed vocabularies and techniques for associating concepts. And they have a very nice website where you can search for a term and see if what are the communities that are using this term for, and how they relate between them. And for example, if you are using, you don't know, for example, sample frequency, well, this is a very basic term, but you will say, okay, Bits uses it in this file, this other vocabulary like Artemis uses it in another way. So you can see how the same term links between vocabularies, and also allows you to search across data sets. data management is really important because many files change over the course of a project. So we need we need a way to smartly track them. And precisely, Data Lab is an excellent tool to do that. And it allows you to manage and version control digital files in a decentralized way and tracks data and metadata of any type and size in a scalable Git repository structure. It can be stored, data can be stored on local devices or remotely or on the cloud. And it's really easy to retrieve the data via some commands, regardless of where it's stored. And it can work with many major providers like Open Neuro or the Canadian Open Neuro platform, Indie, or any other. As long as you have permission or you provide authentication, data can be easily recovered. Here is a very nice schema on how it works. I recommend you to go and have a quick look and start using it because it's really useful for tracking your data and the different versions. And then we have BrainLife, which is an open non-commercial cloud platform that allows reproducible data management and analysis, and it automatically tracks data provenance automatically. And it allows you to manage your data, your processing apps, and your derivatives into one single platform. It's really user-friendly, and it allows visualization and the publication of your results. Here is a very general schema of the parts of the components it has. You can upload datasets from your own or from Open Neuro and other open repositories. You can then archive them and try to run some processes through apps that you can. There are many available apps, but you can also develop your own apps and upload them to the platform. And there are many servers and resources available, freely available for use for everyone. But also you can have your own resources. If you want your data doesn't leave your computer, you can add your own resources. And if you want the apps run on your server, then you also can add your resources there. So you can run apps, then visualize results, archive them, and finally publicate them on the website. So this is a very useful and integrated way to have everything in one place. And this is in BrainLife.
Cool, thanks, Guiomar. So yeah, the next part will be about data processing and analysis, and then we'll do the research disseminations and we'll summarize everything. So for the data processing and analysis, there are so many open tools that are available. For MRI, for example, the three main tools that people use to process their data are usually FSL, SPM, or AFNI, and all of them are open source. So you can use all of them and know exactly what's going on. And there are also other more specific types of software like FreeSurfer, ANT for registration, and iBible to read and write images in some formats. You can use NiLearn if you're doing more statistical analysis, visualizations, and machine learning. For EEG, you have EEG Lab, Brainstorm, MNE Python, and Fieldtrip. So there are really a lot of software to use that is open source, which is great. And there are also some workflow engines. So when you have, for example, you can use a specific software to do pre-processing and then another one to do your GLM in MRI or something else. And then you need to connect them sometimes somehow. So you can use some engines to do it. You can either write your own scripts and code, you can do bash scripting, you can write in whatever language you usually write, MATLAB, Python, R even. Or there are some more neuroimaging specific libraries in Python, for example, like NiPipe, which is very useful for general purpose scripting. There are major attempts to standardize workflows and neuroimaging, and there are some really prominent examples for this. So CPEC, for example, the configurable pipeline for analysis of connectons, is like a nose to tail pre-processing and analysis pipeline for resting state of MRI. which just allow you to configure what you want to put in the pipeline. fMRI prep takes a different approach, trying to not just give you all the options, but make like the ideal or standard option and then let you play with it a bit. I think fMRI prep is very, very widely used in MRI, including pre-processing of fMRI. And they also develop some other night preps, it's called, so other neuroimaging pre-processing tools for example, diffusion and for other types of data. So this is very useful to use, to standardize how we work and have use the benefits of all softwares together in one workflow. And there's also the M&E beads pipeline that can be used if you're doing that. There's also some tailored workflows that have been done by the Large Scale Acquisition Initiative, like the Human Brain Connectome, for example. They just publish their own software, and you can use it with their data, and you can do a lot of different analysis with them. This is also very good and open source. And all these tools increase the transparency and the predictability and the automation of the analysis, which is very useful for all of us. And many of these tools also use containers, which are kind of a virtual machine environment you can run on your computer that is independent from your specific computational environment, but it uses the computation environment that is needed for the tool. So you have already all the packages, all the right versions, everything you need in one place, and you can just run fMRI prep as a container in Singularity or in Docker. And this is very helpful to do it more smoothly and more easily. Of course, sometimes it's not enough, all these great tools, and sometimes you want to do different analysis, or for other reasons you need to develop your own software implementation, write your own code. And one problem with this is great, but one problem is that most of us in neuroimaging were not officially trained, formally trained as software developers. And sometimes we don't have, we don't know the best practices on how to write code. And this is really important to make sure we don't make errors and that people can reproduce our code later. So some good practices that would be good to implement your own programming is, first of all, version control. And the main tool to do this is Git, and there's the web site GitHub that you can use with application GitHub. But you can just, so version control means you have your code, exactly each version that was changed. You have a message about it, commit message about what was changed. It tracks exactly what was changing in version, how you can go back if you need, you can fork, and you can do a lot of things with that. And this is really, really, really helpful. Dataled also helps with that, like Guiomar said before, and it can also be used in for larger files, because Git has a limit of the size of file you can have. So imaging data is sometimes does not work with GitHub, but it could work with Dataled, which is based on Git. You also need to make sure you document your code correctly. So one thing I should have said before is that this helps when other people want to use your code to do their analysis, to adapt to their studies, or to reproduce your analysis. But it also helps you, because I don't know the source of these citations, but I really like it. So I'm going to say it. It's that your primary collaborator is yourself 6 months from now, and your past self does not answer emails. So, if you go suddenly you need to revise something, you have a revision in your paper, or you just need to use the analysis for something else. And many times you just don't understand what's going on in your own code after a while. And making it, version control, documented, test everything, is really, really helpful to make sure you can use it again and other people can use it again, and that it's better and is less likely to contain errors. Testing is something that is not done enough for sure in neuroimaging. You need to make sure if you enter, insert some kind of input, you get a reasonable output, and make sure that things are working properly. If you can, it really helps if someone else could do a code review and look at your code. You can walk them through, and then you can see also if it's clear or not, what you can document better, if you can give better names for your variables, and find errors like that, and that helps a lot. And at the end, it's really helpful to share your code, but this will cover in the next section. For the statistical modeling part, Guiomar talked a lot about bits and how we name the different files and how we put them in the different folders. Which is really, really important and made a huge advanced neuroimaging considerably. But one problem is that at the model specification level, we didn't have a format standard that is used across labs. So this is being developed now as part of an extension to BIDS, a model specification for GLM. It's called BIDS Stats Model. It should be released very soon, if it maybe it's already been very recently. And it's just a structure of a JSON file that tells you how to write the different parts of your GLM, general linear model, so that machines can read it, software can read it, but also people can understand it and know exactly what model you did, participant level, scan level, group level, everything. Which contrast, which regressor, which covariance you use. It's a machine readable and human readable. It's really, really, really great. So keep an eye for when it comes out. And there's some software that can use this to fit your model, like FitLinks that's been developed based on AFNI. And also NeuroScout is a website that uses beat stats models and FitLinks to do reanalysis of a lot of different data sets for naturalistic stimuli. So this is an example of how it can help us automate a lot of things and do it better. So I again encourage you to test it, try it. And like I said, there are so many software, so many choices you can make, which is great, but it's also concerning because these methodological choices that could be arbitrary in a way, not all. Many times there's not like an optimal choice or a clear choice on what you need to do, which software to use, what model to use, how exactly to do the pre-processing and all this kind of stuff. And when you just choose kind of randomly or arbitrary between different good options, this could affect the results. So already in 2012, CARP published a paper where it took a single fMRI data set and just ran almost 7,000 different pipelines with it. And he showed that some outcomes were quite stable across different pipelines, but some of them were really dependent on a specific pipeline that was used. And 2 years ago, we published a paper where we took it to the next step, and we collected new fMRI data set and we distributed to 70 independent analysis teams around the world and asked them to, here's the data, here are 9 predefined hypotheses, please use the data to test this hypothesis, however you usually analyze data, MRI data in your lab, and report back, the result for each hypothesis and bring us the statistical maps. And what we found was, first of all, extreme flexibility in analysis. So 70 different teams chose 70 different pipelines, which was really, really, I don't know, surprising or not, but quite extreme. And this did affect results for many hypotheses. So for some hypotheses, there was convergence, but for some hypotheses, different teams that made different choices also got different results and conclusions, which means that it does matter, all these different choices that we make, and we need to be conscious about it. And one problem with that is that, we already described a lot of awesome tools for open and reproducible neuroscience, but all of these tools will not solve this problem specifically. Because even if you, ideal situation, you pre-registered your analysis, you followed it exactly, you shared your data, you shared your code, everything is documented, no errors, everything is just great. You still, we don't know what would have happened if you did, something else. If you chose a different software, you chose a different model, but pretty similar. And this is a problem because we don't know how robust your results are to the different analytical choices you made. So what we and others are suggesting is to use a multiverse analysis, in the future, where we take the same data and hypothesis and we test it with different pipelines, and then we look for the convergence. Want to see which results are robust to the different analytical choices, or maybe which are not, and we can learn from this as well. This is of course hard, both practically, computationally, and also conceptually. How we converge across these results, how we report them, right? It's really hard in one paper to report this many. So there's some progress that's being done, and a lot of work that's being put into this. So hopefully we will have solutions for this soon for MRI. There are already really great solutions for more simpler data, like behavioral data, or just when there are less analytical choices, or where the result is not a statistical net, but just one result, which is easier. But a lot of awesome people are working on this. So hopefully we can start using tools very soon. And now take it back to Guiomar.
Yeah, so finally we reached the research dissemination section. So as we have seen throughout the research cycle, we produce multiple outputs far beyond publications, and each can have different levels of reproducibility and different levels of openness. So as we can see in this figure, which is from the Turing Way, which is an awesome initiative for reproducibility of research. We can have data, we can have code, some tools, some results. All of these should be shared following the FAIR principles, which is data and code and results should be findable, accessible, interoperable, and reusable. So findable, what does it mean? So findable should be, should use persistent identifiers like POIs and rich metadata that can describe your data and be indexed in a searchable resource. So sometimes, okay, you have in it, it can be institutional or it can be open, an open resource, but it should be somewhere that people can easily access and find your data. Then accessible, share, as these two are often very related. It should be shared in a public resource, which open or control access depending on the regulation. So sometimes if the regulations doesn't allow to be fully open, it's okay, it can be still public, but with control access for specific people. And then it's important that data and code and everything or any research you share is interoperable. For this, it's important to follow a common standard for organization and vocabulary, like we were mentioning bits, for example. So people can take it and use it again, or use another tools and get some results. So this is also linked to the next one, which is reusable, which is it should be described richly enough, so people can reproduce your results or can reuse it in a different way, and with detailed provenance and appropriate licensing. Licensing is something that sometimes we forget, but this is also very important because it tells you how can you reuse your data, which permission you need or which conditions you have. Then for data sharing, as we were saying, should be fair, and it has multiple benefits. Now it's required by some journals and funding agencies, and as we were mentioning, it can be fully open with uncontrolled access, but it can still be public with control access, and it's also good depending on the regulations of your country or institution. There are many free neuroimaging data sharing platforms, specifically, as I'm saying, specifically dedicated to neuroimaging, like Open Neuro or Brain Life or Gene, Dandy, Indie, NITRC, NDA, the Canadian Open Neuroscience Platform, eBrains. All these platforms allow you to share your neuroimaging data, but still there are other dedicated repositories, for example, the UK Biobank or HCP, CamCam, Omega, IBCD. For example, the CamCam is a collection of data that contains MRI, MEG data, and it's fully dedicated to a project. So these are also good examples of repositories where people have shared their data and that you can nicely use it. And as well as this, there are generic archives, like they are not neuroimaging specific, like OSF, Zenodo, GitHub, or Fixate, where you can also share your data there, even though they are not neuroimaging specific. Together with the data is really sometimes very recommended to use published data descriptors, which are not intended to communicate findings, but detailed information about the dataset, how it was collected, what includes, how to be used, and along with the shared data. And there are some initiatives that are starting to emerge to recognize data sharing efforts, like the Open Science badges in journals and prizes like the OHPM's data sharing prize, which at least recognize a bit the data sharing efforts that sometimes are not as well incentivized in the research careers. And sometimes people say, okay, I'm going to share my data, but what's the benefit of that? Well, we should know that there are many benefits, but maybe they are more long-term. For the methodological transparency, how you share your code and your scripts, as we were saying, documenting the analysis steps is key for reproducibility, but sometimes the articles, and paper method sections is often not enough. It's short, you can share a bit of what you've done, but at the end of the day, code is the ultimate source of the exact analysis you followed. So this is really important to openly share, so everyone can reproduce or see what you did. And it's important to bear in mind that sharing imperfect code is still much better than not sharing at all. So even you are fearful that, okay, it's not very well organized, or okay, it's fine, but at least share it so people can see. And sometimes we also use GIS, which is a nice and interactive way of analyzing your data, but it can be a bit sometimes challenging to report the steps followed, even though some GIS or some toolboxes that work through GIS often some data history and provenance. This is really useful. So otherwise, clicking buttons sometimes it's more difficult to report what you did exactly. And the importance, as we were mentioning throughout the talk, of having version control options of your code and permanent DOIs, which allows you to store your scripts and ensure that it's not going to change or disappear in a few years. So we always try to use platforms that allows you some more permanent storage. And finally, derivatives, which is really critical, is how you process your data and how you obtain your results, your final outputs. Still, it's really challenging because today there are not many available or mature specifications for advanced derivatives. So there are not that many standards, for example, for connectivity, so the community lacks a clear guidance and tools. There are some efforts still there are some efforts, and we have to continue pushing them. For example, WITS has released a general specification for common derivatives throughout all the neuroimaging modalities, and the community is actively working for extending and advancing derivatives for the different other more specific neuroimaging modalities. So I recommend you also to have a look at WITS derivatives as a starting point. Related to MRI, there are a few open neuroimaging repositories like NeuroVault, which can store statistical maps and parcellations, and atlases from MRI and PET, or template flow for MRI templates and spatial maps across standard species, and BrainLife, which can integrate and process apps and derivatives. And well, we have to bear in mind that also table figures and slides should all be shared fairly, and because they are also part of the derivatives and results and outcomes of your research. And finally, scientific papers, which is like the historically most important means for disseminating your results. There have been important initiatives and community efforts for guidelines and best practices, like OHBM COVIDAS for MRI, MEG, and EEG, or guidelines for PET, or the Artemis EEG reporting template, which allows you to see, okay, how to properly report your analysis and your results. And also, it's important, I think most of us are aware of this problem, that papers should be accessible to everyone. Sometimes scientific publications are hidden behind paywalls. And even though this is slowly changing, with both researchers and funding agencies pushing towards open access, however, the adoption of how this open access is implemented by major publishers may be considered arguable, like pricing or tracking, but still we should, that's why a good option is to release preprints, which are prior to peer review in scientific journals. You can openly share your manuscript, and this is shared immediately, so it can accelerate the release of new findings and allow early feedback prior to final publication. There are initiatives for reviewing preprints, like review or the Open European Research, which is an initiative to share openly your preprints. Also, if you have done research funded by Horizon or Europe. And also there are some hybrid publication formats that can include text, data, and runtime environment, which is very useful because you can publish your paper together with your scripts and to your results, and it can run. You can run it all together, and it's all released together, like Neurolibre. And some traditional publishers have partnered with companies like Code-Ocean, and they are having also very nice results. But Neurolibre is an open initiative. Well, as we have seen, the research life cycle continues beyond paper publication. Dissemination and outreach are really important parts of this. There have been a shift, rather than carrying out a study from inception to publication, from inception to results, and only then releasing the finding and disseminating findings. Now researchers have a lot of opportunities to share their ideas and the results as they are being developed, like with standard open procedures, with preprints, with pre-registration, and all these steps that we have been reviewing in the talk. So more transparent, inclusive, and collaborative processes that should aim for equity, diversity, and inclusivity in our research. So yeah, finally, as conclusion, sorry, my apologies. I took COVID yesterday. I tested positive, so I'm a bit, my voice is not that perfect, but yeah, as a conclusion, there are abundance of tools and practices, open tools and practices, which is really promising and challenges, but sometimes it's difficult to realize the whole schema. So we have tried to provide an expert-driven integrated review, and you can check the preprint for much more information. And we have also, in the preprint, you will see an additional material and a resource table with additional material links for at every step of the research cycle. How can you really make it real from the theory to the practice and implement this in your research? Thank you very much.